Look at the back of your own hand for a second. Ordinary skin, ordinary veins. But inside almost every one of the tiny cells under that skin, there is a complete set of written instructions for building and running the entire living you. And nearly all of those instructions are stored in a single spiraling molecule. Your eye color, your blood type, the shape of your face, the way a cut on your skin knits itself back together, the recipe for all of it is coiled up so tightly that it fits inside a space far too small to see. And this is not a figure of speech. It is a literal chemical text, written in a 4-letter code, sitting in your cells at this exact second, being read and copied while you listen. Today, this is the most famous fact in all of biology, and you have seen that twisting ladder 1,000 times on posters, on the news, on the side of a science kit for kids. But here is the part that should stop you. There was a moment, less than a lifetime ago, when nobody actually knew which molecule carried her pinty, and the person who proved it was so quiet and so easy to overlook that the world handed the credit to other people and let his own name go quiet. His name was Oswald Avery, and for most of his life, he was a careful, unassuming researcher who studied the bacteria that caused pneumonia. He spent his days at a microscope chasing one stubborn question, and the answer he found turned out to be one of the deepest facts about your own body. Working with 2 colleagues, he showed that the instructions for life, the whole living blueprint you inherited from your parents, are written in that one molecule, and not in the one almost everyone expected. He published it a full 10 years before that famous ladder was ever drawn. Now, I want to be honest with you from the start, because the truth here is better than a simple tale of a robbed genius. This was not a solo act, and the doubters were not all fools. But when the dust finally settled, the paper he and his colleagues wrote was called one of the most important in the history of biology, and the Nobel Prize went to other men. The podium meant for him stayed empty. So let me show you exactly what he found, why so many brilliant people refused to believe it, and how the credit for one of the deepest facts about you drifted quietly away from the man who found it. Silent subscribe plus like reveals here at end of board 1, visual only, leaning 3s, no narration mention, rule 28. Let's stay with your own body for a moment, because the scale of this is genuinely hard to believe. You are built from something like 30 trillion cells, and almost every single one of them carries its own full copy of your instructions. If you could pull the coiled molecule out of just one of your cells and stretch it straight, it would run about 2 meters long, taller than you are, and yet it packs itself down small enough to hide inside a speck you could never see. Think of it as a recipe book for the whole of you, written in a chemical alphabet of just 4 repeating pieces. That book decides the color of your eyes, the pattern on the tips of your fingers, the resemblance that makes a child look like a parent. And here's the working part you feel every day. Every time one of your cells wears out and splits to replace itself, it first unzips that entire molecule and copies the whole recipe, so the new cell inherits the same complete instructions. It is happening in you by the millions right now as you listen. Put a hand on your arm for a moment. Under your fingers, cells are dividing, and inside each of them, that whole 2-meter recipe is being unzipped and copied, letter for letter, without a single mistake most of the time. That is the everyday miracle sitting quietly inside you. And for the longest time, the one thing science could not answer was the most basic question of all: which molecule actually holds the words? So the molecule is astonishing, but here's the twist that makes this a story. For a long stretch of the last century, even after scientists knew this coiled molecule was sitting there in your cells, almost none of them believed it could be the thing carrying your heredity. And their reasoning was not stupid. Look at what a cell actually holds. It is packed with proteins, which are gloriously complicated, long chains folded into intricate shapes, made from 20 different building blocks, able to do almost anything. Next to that, the coiled molecule looked boring. Just those same 4 pieces, over and over, in a plain, repeating ladder. If you had to bet on which of the 2 carried the entire instruction set for a living thing, the smart money was obviously on the rich, clever protein, not the dull, simple ladder. How could something that monotonous possibly spell out a whole human being? For decades, that question just sat there, unanswered, and the easy assumption won, until a quiet man studying pneumonia decided to actually put it to the test. Oswald Avery worked at the Rockefeller Institute in New York, and by every account, he was the opposite of a showman. Small, soft-spoken, endlessly cautious, he hated overstating a result, and would sit on a finding for years until he was certain of it. His subject was the bacterium behind pneumonia, which in his day was a leading killer. And that germ came in 2 very different forms. One had a smooth, sugary coat that hid it from your immune system. That one was deadly. The other had a rough surface with no coat, so your body mopped it up easily. That one was harmless. Smooth and lethal, or rough and harmless. Which form a germ was seemed fixed, like a trait you're born with. Then, a few years earlier, another researcher named Fred Griffith had run an experiment with mice that made no sense at all, and it was that impossible result that Avery could not stop thinking about. Here is what Griffith saw, and you can follow every step. Inject a mouse with the live deadly germ, and the mouse dies, no surprise there. Inject the live harmless germ, and the mouse lives. Now take the deadly germ and kill it with heat first, then inject that, and of course the mouse lives, because dead germs can't hurt anything. But then Griffith did the strange thing. He mixed the dead deadly germs together with the live harmless ones, two things that on their own could not kill a mouse, and injected the mixture, and the mouse died. When he looked at its blood, it was teeming with live deadly germs. Somehow the harmless bacteria had been permanently transformed into the deadly kind, and their offspring stayed deadly ever after. Something had passed out of the dead cells and into the living ones and rewritten what they were. Griffith called it the transforming principle. He had no idea what it was made of. That unknown substance is exactly what Avery set out to catch. Avery did not do this alone, and that matters. He worked shoulder to shoulder with two colleagues, Colin MacLeod and Macklin McCarty, and the three of them share the credit for what came next. Their plan was simple to say, and brutal to do. If some substance could carry a heritable trait from dead cells into living ones, then they would grab that substance, purify it until it was clean, and find out what it was chemically made of. So they grew bacteria by the bucketful, burst the cells open, and began stripping the soup down, step by patient step, until they held a thick, stringy extract that still carried the transforming power. It took them years. Avery, cautious as ever, refused to announce anything until the case was airtight. And to make it airtight, they built a beautifully ruthless test, a way to identify the mystery molecule, not by looking at it, but by destroying its rivals one at a time. Think about how patient that is. They could not simply hold the substance up to the light and read its name, so instead they decided to work like a detective in a locked room, removing suspect after suspect until only the culprit was left standing. It is one of the most elegant experiments you will ever hear described, and it turns entirely on a single, ruthless idea. Here is the logic, and it is gorgeous. Whatever the transforming substance was, it had to be one of a handful of molecule types in that extract. So they used enzymes, molecular scissors that each chop up one specific kind of molecule and nothing else. First they added the enzyme that shreds proteins, the very molecule everyone assumed was the answer. If protein carried the trait, the transforming power should now be gone. It wasn't. The extract still turned harmless germs deadly, so they kept going. They destroyed the sugars. They destroyed the fats. Still it worked. One by one, every suspect was crossed off, and the transforming power just kept going, until they reached the last candidate and added an enzyme that chops up only one thing, the coiled molecule, DNA. And the moment its DNA was shredded, the transforming power vanished completely. No transfer, nothing. Every other molecule could be destroyed, and heredity still passed, but destroy the DNA, and it stopped dead. There was only one thing left it could be. Sit with how clean that is for a second. They did not have to argue about it, or guess, or appeal to anyone's opinion. They let the experiment answer. Every rival molecule could be torn to pieces and heredity carried on regardless. Take away the DNA, and the whole thing collapsed. The molecule everyone had written off as too dull to matter was the one holding the words. In 1944, Avery, MacLeod, and McCarty published what they had found. The dull, simple molecule everyone had dismissed, the coiled ladder, was the transforming principle. It was the material of heredity, not the protein, the DNA. If that sounds to you like it should have set off fireworks, you would think so. But this is one of the quietest bombshells in the history of science. Avery, being Avery, wrote it up carefully, almost under his breath, hedging every claim. Around the same time, he sent a long letter to his brother, laying out what they had seen. And even there, in private, he wrapped it in caution. He knew exactly how big a claim he was making, and exactly how easy it would be to shoot down. He had, in effect, just answered a question people had argued about for decades, and instead of celebrating, he braced for the fight. And the fight came because a lot of very smart people simply did not buy it. Now, here is where I have to be fair with you, because it would be easy and wrong to paint the doubters as villains. They had real reasons. Remember, the DNA molecule still looked far too plain, just 4 pieces repeating to spell out something as rich as a living creature. The complex, versatile protein still felt like the obvious home for information. Asking people to believe that plain repeating ladder held the whole recipe was asking a lot. But the strongest objection was not snobbery, it was good science. When you purify a substance out of a living cell, you can never be totally certain you removed every last trace of everything else. What if a tiny invisible smear of leftover protein was clinging to that DNA, and that was the real active ingredient? Avery's extract was mostly DNA, yes, but was it the DNA doing the work or a contaminant? It was a fair question, and an honest one. So the discovery landed with no fanfare, the doubt hung in the air, the paper sat on the shelf, and the years began to pass. The loose end that hung over Avery's work, the worry about that trace of protein, was finally tied shut a few years later, and by someone else. 2 researchers ran a now famous experiment using a virus that attacks bacteria by landing on the surface and injecting its material inside. They tagged the virus's protein coat and its DNA with 2 different markers, let the virus do its work, then spun the whole mixture in an ordinary kitchen blender to shake the empty virus shells loose. When they checked, the DNA marker had gone inside the cell, and the protein marker had stayed outside on the shell. The genetic instructions were carried by the DNA. It was exactly what Avery had told everyone years before. And almost right after that, 2 more scientists worked out the shape of the molecule itself, that elegant double helix you know so well, and suddenly it was obvious how a simple ladder could store endless information. The world finally believed. And here is the quiet cruelty in the timing. By the time everyone agreed, the argument felt settled by the blender experiment and the double helix, the shiny new results people could point to. The careful old paper that had said it first, years earlier, slid to the back of the story. The trouble is, by then, Avery's role in it had already begun to fade into the background. So picture where that leaves him. The man who first proved which molecule carries heredity is now an older, retired researcher, watching a field he cracked open get crowned with the highest honors in science, and the Nobel Prize never comes to him. And I want to be careful here because this is not a story of theft or a villain. Nobody stole his work. He was nominated for the prize many times over, but the committee kept hesitating, partly because that old doubt about the contamination had made his result look uncertain for so long, and partly because science, like people, is just slow to admit when it was wrong. The chair meant for him at that ceremony simply stayed empty. In 1955, Oswald Avery died, and the greatest prize in his field was never put in his hands. His name, and the names of MacLeod and McCarty beside him, slid quietly toward the footnotes. But the footnotes got it wrong, and time has been slowly setting it right. That careful hedged little paper is now regularly called one of the most important in the entire history of biology, because everything came after it. Once you know the recipe lives in the DNA, you can start to read it, the whole genetic code that turns those 4 repeating pieces into a living body. Every modern biotech lab, every gene-based medicine, every crime solved by a genetic match, every spit-in-a-tube ancestry kit that traces your family back through the centuries, all of it stands on the fact that Avery and his colleagues proved first. None of that works until you know the instructions live in the DNA. He did not just win an old argument between 2 molecules. He handed us the thread that the entire modern story of life would be pulled from. So go back to where we started. Look at the back of your hand again. Inside almost every one of those cells is that same coiled molecule, holding the whole instruction set for you. You already knew it was DNA. Now you know the quiet man who proved it. His name was Oswald Avery, and the credit belongs to him and to MacLeod and McCarty together. The prize never came. The truth did anyway. So the next time someone shows you that famous twisting ladder and calls it the most famous molecule in the world, you can tell them who first proved what it really does. If Avery's story stayed with you, do me one small favor. Like this video, subscribe to follow the rest of these, and tell me in the comments whose forgotten story I should draw next. There are so many of them still waiting for someone to finally say their name.